Hi everyone, really happy to be here. Okay, so let's talk about pigments. Pigments are what make our world colorful and beautiful, and pigments are found throughout all kingdoms of life. Now, pigment cells function either by absorbing light or by reflecting and scattering light. And these pigments that reflect and scatter light are called structural colors. And they have many functions, such as camouflage in the chameleon. We have vision, like in the eyes of the scallop and the zebrafish, and communication, like the really cool copepods, and also for the zebrafish. And all these examples that you see here today are actually organic crystals that are made of guanine. And this is the same guanine, the G, that we have in our DNA. Our lab, the Gur lab, is interested in these crystal-forming cells. And more specifically, we're interested in the crystal-forming organelle. And to study these cells, we utilize the zebrafish, which are a really cool animal model. And they have 3 different types of pigment cells. We have the xanthophores, which are yellow, the melanophores, which are black, and the iridophores, which are these structural colors, which are light and shiny and reflective. Now, so for my talk today, we need to be more familiar with the 2 types of pigment cells. We have the black melanophores, and they absorb light. And they have the melanosome. This is the pigment that has the melanin. Melanin is the black pigment inside of him, and we humans, we have this pigment. We have melanin. And on the other hand, which is the exact opposite, there's the iridophore. The iridophores reflect light. They have the iridosome. This is the organelle that contains the guanine crystals inside of them. We humans do not have iridophores. Unfortunately, we're not shiny. And some important genetic markers just to remember: for the melanophores, we have TIRP1B, and for the iridophores, there's PNP4A. Now, really interestingly, the lab of David Paricci, which also study pigment cells, they discovered a new pigment cell in the zebrafish, and it was shown to be something in between a melanophore and an iridophore. So who are these cells? These cells are the melanoleucophores. It's a big name, but once you understand it, it's not too bad. And the melanoleucophores are found in the distal edges of the dorsal fin and also in the tips of the tail. And they were shown to express both the iridophore marker, the PNP4A, and the melanophore marker, the TIRP1B, which I just mentioned. And probably the most interesting aspect of these cells is that they perform some sort of transdifferentiation. So they start off as this black, brownish cell, and they shift into this white, shiny, and reflective cell. And the Paricci lab did an amazing study to discover and characterize these cells, and they showed it mainly on a genetic level, just by looking at the genes with single-cell RNA-seq, and we can see that they start from melanophores and they shift into melanoleucophores. And this was just by looking at their genetic expressions. But what we don't know is what these cells actually look like on the structural level. And this is what I'm going to talk about today. So what does transdifferentiation look like? So we're asking, what is the cell ultrastructure? What's the crystal formation mechanism and the crystal composition? And what's happening with the melanin? Because we see it disappearing, it's degrading, and whether these processes are connected. So due to lack of time, I'll only speak about the 2 top ones. So the first thing we wanted to do is to understand when this happens. So we followed the fish, and we saw that that transdifferentiation initiation is when the fish are juvenile, they're around 4 weeks old. And then we can see a strong increase in the number of these cells. And then we can pinpoint, and this was like the guiding pillar of when this happens. So we can then look before, during, and after transdifferentiation and further understand about this process. So the first thing we wanted to know is, so what do the fully differentiated melanoleucophores look like in the adults? So this was with the help of Neta and Katya from the electron microscope unit, and with a special electron microscope that has the slice and view method, we could visualize these cells in their native state and get a full 3D image. And I hope you can appreciate that in blue, we see that these cells are packed with crystals. And this was really amazing to see. What we wanted to know next is what are the crystals made of? So what's the crystal composition? So with HPLC, we could see high levels of guanine and hypoxanthine, and then we could see these metabolites on mass spec imaging, so we can see the spatial information. So I hope you can see in green and in red, this is guanine and hypoxanthine that are found in the tip and the edge of the fin where these cells are located. So this was really interesting to see both the composition and their localization. Okay, so what we wanted to do now is to be able to isolate these cells to go to higher magnification methodologies of cryo-EM. And for this, we needed to generate a double transgenic line for sorting. So we took, in green, we have the iridophore marker, and in red, we have the melanophore marker, and this is what the fish fin looks like. So we have these two colors. And then we could go on and digest the tissue, sort them with FACS, and head over to higher magnification methodologies, such as cryo-TEM, which is, if you don't know, a great electron microscope that really gets you inside these cells. So what we could ask is, so what do these crystals look like? What is their morphology? And the first thing we notice is that each crystal is engulfed by a membrane. They were all membrane bound. The second thing we noticed is that they had so many different shapes and sizes, kind of like snowflakes, what we think of them. We couldn't see two crystals that looked the same. And this randomness in morphology got us really interested in the mechanism and the crystal formation mechanism. So for this, we went to earlier stages of the fish, as I mentioned earlier, so younger fish when this crystal formation mechanism is just happening. And what we saw is we had vesicles that contained randomly oriented fibers. And on top of these fibers, we could see nucleation. So this is termed templated nucleation on top of these fibers. And inside the same vesicle, we could see multiple templated nucleation events, and then crystal growth. So this explains to us why there's this randomness in morphology. And this was with the amazing Zohar Eyal, which is also here with us. And Zohar also published a paper recently in our lab that showed the same kind of idea, the crystal formation mechanism in iridophores. And we see kind of the same thing, that we have these fibers and templated nucleation on the fibers, and the crystal growth. But the crystals have different morphologies, which is interesting to us in the lab. And to make this even more interesting, if we compare it to how the melanin is deposited in the melanosomes, it's also on fibers, and there's melanin deposition inside of these organelles. So this was really interesting to see this fiber motif kind of coming up in all these different pigment cells. Okay, so what we wanted to know now is, so what does it look like? What does the transdifferentiation look like at the ultrastructure level? Can one cell actually have two different pigments? Is it happening in the same time? Is it compartmentalized? What's happening here? And again, using the FACS and the cryo-TEM, we could separate them, and we could see fully differentiated melanophores. Now, I just want to make it clear that we're looking inside a single cell. So all of these black-shaped round thingies, this is the melanosome. This is an organelle that has the melanin pigment inside of it. And then we could see the melanoleucophores that have all these crystals inside of one cell. So we have many, many, many organelles inside a single cell. But what was truly amazing is that we were able to see two different pigments inside a single cell that is going through transdifferentiation. And this means that these two pigments are there in the same place in the same time, and this is one cell that's doing it. So if you think of it, it's actually one cell acting as two different cells. So this was really cool for us to be able to see and visualize what this transdifferentiation looked like. And so the last thing we wanted to know is this the same process? Meaning, or what's the connection here? Is this the melanosome, the melanin organelle, that's shifting gear and then changing into a crystal, or we have completely two different things happening in the same time? So for this, we utilized the cryo 4D stem, which is a big word for an electron microscope together with spectroscopy. And the spectroscopy gives us the elemental composition of the substance. And here, together with Lothar, we could visualize a cell during transdifferentiation, so a cell that has both types of pigments. And we could see that in the crystals, they were abundant with nitrogen in green, because guanine is packed with nitrogen. So this gives us this validation. And then with the melanosomes, we know that the melanosomes are enriched with zinc. And what we noticed is that we couldn't see any overlap. We don't see any remnant of purple or zinc inside the guanine crystal, meaning that it's probably a different organelle, and there's no leftovers of this organelle in the crystals. So for this, I'd like to summarize and show our proposed model. So we first saw that they're irregular shaped crystals, and they are all membrane bound, meaning there's a membrane around each and every one of them. And we saw the crystal formation mechanism, so we see it's through templated nucleation, similar to the iridophores and the melanophores. And we see that melanin degradation and crystal formation are happening in the same place in the same time, but they're different and they're separate processes. And with this, I'd like to thank everyone that's helped me and been so supportive, and for you to listen until... Thank you.